Try to escape these spotlights here by moving around and keeping you awake. Thank you very much, James, for that kind introduction. I do indeed remember the three of us sitting in a conference room scheming out Regenix, which was pretty exciting, and look where you brought it. Great as well to work with you as well as with Aldevron and several other folks here in the room. So I will talk to you about a number of things, but before that, my conflicts of interest, which again reflect kind of where the space is going, kind of an integration of where a lot of this started, academia, with biotech and pharma. So take everything with a grain of salt that I'm telling you. So the common denominator of the presentation I'll give you is basically broadening the reach of gene therapy and our efforts in that space. I'll highlight two different aspects of that. One is our approaches towards discovery in terms of getting to that broadening of the reach of gene therapy, as well as in a second, smaller part about how we could do this through setting up collaborative networks. Gene and cell therapy, and I'll be mainly talking about in vivo gene therapy, are often seen as kind of this boutique space in terms of drug development that really is very unique. But I think it's a healthy perspective to really see that fully as a space within drug development where there's actually more analogies than there are differences. And let me highlight how I, maybe in a simplistic manner, look at this. I mean, every drug discovery effort really is the bringing together of a target, and again, that could be a molecular target, but also as a cell target or a tissue target. But a molecular intervention at such targets that intervenes with a disease process. And then what's often forgotten is the delivery component. One needs to bring such intervention safely and appropriately towards that target. Now, the only space really where gene therapy is different is that that delivery component is done for a gene or a genetic-based product. So in that sense, many of the other aspects are really analogous. Now, a step back in terms of where we are historically and how we got to this exciting moment in time where we can have the fortune to look at clinical trials moving forward and even products emerging on the markets, is really that we see this progression of science on the bottom side of the slide in lockstep with these innovative ideas towards drug development, where we started decades ago in the century, if you will, in terms of small molecule development, the onset of molecular biology, our ability to sequence and our understanding of genetics, as well as some understanding of developmental biology, and now, and we've heard already this morning, the advent of the tools that synthetic biology brings allows us to make more and more sophisticated drugs, and that brings us to the space we're at now in terms of cell and gene. So what that does to this chart is that the number of targets that we can explore, the various ways we can intervene in disease at a molecular level, as well as the delivery modalities, are just exploding. And that's actually a challenge to the field, and that's actually one of the reasons why the tool set and the abilities of an Aldevron of this world allow us that flexibility to work with all of the. One of the key things here is that because of this explosion, we're also dealing with increasing demand on the gene deliveries. Because each of these targets, each of these interventions require a indication or modality-specific delivery. And I'll be mainly talking about in this first part about how we think about delivery. Before I go there, why do we even need cell and gene therapies? I think I don't necessarily need to convince this audience, but really there is still a significant unmet. You classify that broadly. There are undruggable, with a single D, undruggable disease targets, the single gene disorders, but those expands to more common complex disorders. And then there are remaining challenges in terms of delivery, whether it's on the pharmacological side, getting steady state delivery, or hit and run kinetics, whether it's specificities. All of these things we can dramatically improve and currently prevent us from adequately treating certain. So it's really these technology gaps, as well as knowledge gaps, that we're trying to address. And again, with the space and the explosion of innovation that's happening in this space, we believe there's a clear opportunity. The opportunity that we are jumping on, as well as obviously the excitement in the space, is around AAV, leveraging AAV, adeno-associated viral vectors, for those delivery. And I will kind of go briefly over this introduction, but AAV has a lot to go for it, kind of out of the box. As a wild-type virus, it has various properties that speak to safety overall. In and of itself, AAV has not been associated with disease, for example. And then we can render this replication defective virus into a vector by eliminating all transgenes and again further adding to the safety of that system. And it's really the safety that has largely driven this vector profile, or this vector, to get to the level where it's at now, where we are indeed seeing the first drug entering the market. And it shouldn't be forgotten that since the mid-90s, we have clinical experience with AAV. Even though only now we see efficacies showing up in trials, since the mid-90s, we have done hundreds of trials, most of which were without significant adverse. Now, another premise to what I'll be talking about next is that we've learned over the decade, working with AAV, mainly in academic labs, that this structure of this buckyball particle, which I'll illustrate in a little bit in the next couple of slides, small variation on that particle can lead to very dramatic, both quantitative as well as qualitative phenotypic. These phenotypic differences initially, since we're dealing with a virus, are viral properties that ultimately translate to really dramatic differences in terms of the translational opportunity. And I'll speak to a few of those. For example, assembly and packaging of a virus directly translates to manufacturing. And we see 10-fold, 100-fold differences between slightly different AAV vectors that have been developed. And I'll speak in a little bit about this aspect that AAVs can sometimes qualitatively jump over physiological hurdles that we never thought were possible. These are large particles. And for example, through a mechanism that we believe is transcytosis, certain AAV serotypes can cross the blood-brain barrier, whereas others cannot. So how have we leveraged that opportunity here, that there seems to be this wide translational set of properties that we can leverage towards bringing new drugs to the clinic? We started in the early 2000s, while at the University of Pennsylvania, with approaches towards biomining. Can we explore more diversity in this AAV? Developed a sensitive PCR method that we then used on primate tissues, both non-human primate as well as human tissues, to isolate an expanded family tree here of different AAV serotypes, different AAV caps. Several new branches in this tree were developed that were previously unexplored, and that actually, I believe, has catalyzed a lot of activity in the space where people took now these new tools and applied them to diseases that were previously unreached. And one example of that, and I think it was mentioned in the introduction, is, for example, this virus called AAV9, which resides in this smaller clade F in the middle on the left there, that then Brian Casper at Nationwide Ohio took in first in 2009, really in just a few years after its initial discovery, and showed that in newborn mice, this AAV9 indeed has the property from a systemic route of injection target the brain and cross the blood-brain barrier. He took that very shortly afterwards to a disease model of spinal muscular atrophy and showed indeed that it could be corrected. And then in a short decade afterwards, has taken this to the clinic for late last year. He presented in the New England Journal data on correction in newborn infants, but really dramatic. And I think you know where this story went further. I believe just 2 weeks ago, this was filed for BLA to now Novartis. So based on these types of results, and there's plenty more of that, although this is arguably one of the most dramatic ones, people have further developed discovery modalities towards exploring that AAV diversity. While we initially started with 6 serotypes, we expanded that to 100 plus through this biomining, and we believe at this stage that these efforts have been overall successful, and the Avexis story is maybe one of those, yet that approach has largely been exhausted. There's been even more creative methods, because biomining really is just phenomenologically exploring the diversity that's out there by engineering serotypes in the lab through a variety of methods, whether they're unbiased, biopanning approaches, or more rational design methods. Remarkably, even though that field has been decades plus old, these are compelling methods, but they have had minimal clinical adoption to date. And in part it is because of the challenges that AAV pose. And where that leaves us now, with the success that we've had in the space, is that the user axis in the space, meaning all of the drug developers in the space, are starting to ask pretty sophisticated questions from this overall very potent tool. And I'll walk you through a few of those that I've received just over the past year. For example, in this SMA example, AAV9 targets the liver with great efficiency, as well as target motor neurons that are relevant to SMA. While SMA is relevant to be corrected in various tissues, and also in liver, probably from a safety perspective, we can dramatically reduce the load to the liver and still have the efficacy. On top of it, it would lower actually the manufacturing. On the flip side, there's other people that want to target liver and that are looking for a vector with more efficiency. Seropositivity, not unlike what was talked about earlier with Cas9, is a key Achilles heel of this technology. Everybody wants to expand their patient base that is currently seropositive and bring these transformative therapies. And again, these questions continue. Just on the manufacturing side, there's questions. We all want higher yields. And in this new era where somebody just invested $20 million to set up a CMC pipeline, when they upgrade their technology, ideally they want to preserve some of it. And then some of these questions that we currently can't even address at all are packaging basically all of these items that are needed for a homologous repair event using Cas9, and ideally with a hit and run pharmacology. Long story short, none of these questions we currently address. And we were compelled to try to provide at least a pathway towards us bringing this technology to address some, if not all of them. So the aims that we have in the lab that I started about 7 years ago was twofold. One is this pre-existing immunity issue is really one that cuts across almost every application using AAV and can cut down between, depending on the studies you read, 30 to 70+% of the population that can benefit. And this is on a human level a very unfortunate event where basically a simple serological assay may tell a patient whether or not he can benefit from possibly a life treatment. And second, to address the questions that I just listed, is there a way that we can start functionally programming these AAVs rather than just an often very unbiased method by just going out to nature or by putting random variation on this capsid? Can we somehow program these capsids to do very specific things that are relevant to the particular application? So how do we think about this? Well, all of these questions ultimately come down to our ability to, what I call here, disentangling different functions of AAV. What I mean with that is that it is a little bit of a Goldilocks type scenario, or another analogy is really a Rubik's Cube analogy, where we're trying to really optimize one facet of this cube and trying to align all the orange colors, say, while retaining the green colors or the white colors that we just spent time aligning on the other hand. Meaning these AAVs have out-of-the-box desirable properties that we don't want to touch, or we may want to improve on slightly, but at certain aspects, such as this pre-existing immunity, such as, say, the hit-and-run pharmacokinetics we want to give it, that we want to modify without touching these other. And that is intrinsically hard. And that is really the challenge that this field faces. The reason why it goes back to the biology of the particle, which is a buckyball particle that is built out of 60 identical units, Homo 60-mer. It's illustrated here on the bottom. On top of that, the architectural definition of this particle is mathematically defined. This is an icosahedron and really hangs together along very strict axis symmetry. So for us to try to probe that complex biology that really gives us very limited degrees of freedom to engineer them, we had this very naive idea several years ago where what if we try to replay the evolutionary path of this virus in the lab? Maybe we can learn from how nature has worked within these limited degrees of freedoms, within these constraints of the architecture, to incorporate all kinds of viral properties that are relevant to its survival, but also, I would argue, are relevant to translation, such as assembly, such as packaging, such as host response. How do we practically do this, or how have we practically done this? Initially, we have tried to reiterate by a computational method the evolutionary path of these viruses by bringing reagents in the lab that are approximations of the various evolutionary intermediates. Really what this is you build the family tree, dendrogram of AAV virus, and you go after these nodes that are highlighted. We do this using synthetic biology tools and computational methods. What we got now 5-6 years ago in a first effort, just as a proof of concept whether this method at all could play, was that we indeed got viruses which are quite distinct. And this was a clear desire because we wanted to move ourselves pretty far outside of the current space, mainly to develop a new antigen, something that antigenically is very. We moved this virus as far as 8.6% from anything that was known in nature. And some viruses are as far removed as 40%. This actually means very practically that we did a mutagenesis effort of 64 positions and remarkably got something viable, as I will show you the next set of slides after. So we dramatically changed this particle and still retained some of these desirable features of biology, which I'll try to convince you. On the surface of this particle, we also dramatically changed it, and actually, indeed, that surface is dramatically changed between 20 to 30%, and this speaks particularly to this issue of pre-existing immunity. And indeed, biologically, we also show that it is indeed a new antigen. When we take high-potency antiserum raised against all of these different viruses, we see that there's orders of magnitude reduced cross-reactivity or absent or non-detectable cross-reactivity between these known. Next question was, does it still work in vivo, which is really where the primary applications, maybe from AAV6 and homologous repair templates, lie. And indeed, what we see here in this first kind of qualitative experiment is that it holds up quite well in a variety of routes of administration, targeting a variety of tissue targets, liver, muscle, retina. And then there were a few surprises which we've worked out and published. For example, out of the box, this AncAAV lead candidate, we call it, actually performs better in this unique property of crossing the blood-brain barrier as compared to AAV9. So on your right here, AAV9 is illustrated, and it may not be all that clear, but this is the basic property that a drug like the Avexis SMA drug build on. Other observations were more qualitative in the sense that all of a sudden we can unlock certain targets, and this is a particular example in the cochlea, where we are compelled to build out hearing gene therapy as a space that has currently not been developed much. What we've shown here is that somehow this reagent allows us to target cells that previously with other technologies we were unable to access, particularly the outer hair cells. If I had a pointer, I would point out to you. But this is a quite robust signal from not being able to target any of these outer hair cells. They're very relevant to many hearing disorders, to targeting 80 to sometimes even 100. So circling back to the aims we set forth, we wanted to develop these antigenically distinct particles. We believe that we have indeed serologically distinct particles. Unfortunately, there's still ways to go, in the sense that the human immune system has a much broader cross-reactivity than what we can model in the lab. And we still see some cross-reactivity with in humans towards even the very distinct antigens that we've generated. So there is still a ways to go in terms of further, if you will, de-immunizing this particle. But I haven't spoken to you yet about how our ability or our hope to program these particles. So how do we want to approach this? And just conceptually, I'll walk you through two of the concepts that we're bringing to this. One is with our ability to have these different generations of viruses being brought alive in the lab, we can now walk lineages. And if you walk one of these lineages, you just see very few changes occurring in each one of these steps. So we basically break down this diversity that we know is phenotypically so interesting into smaller bits and pieces. And those smaller bits and pieces we can now interrogate into the lab. And one of those questions we applied this to is the manufacturing. How can we make more virus? Or can we understand what it is on this particle that drives productivity to be high in certain cases and low in other cases? Conceptually, this is a work of a student in the lab in a more anecdotal fashion. What we did is indeed we took these lineages, we interrogated a particular assembly question, particularly the relevance of a cofactor called AAV. And what we're now looking for is these switch, and we quantitatively assessed this particular assembly function, and we overlaid a heat map on this dendritic. And what we're looking for is the shortest switch point, phenotypic switch, as you can see here between AncAAV and AncAAV. That allows us to fine map a certain set of properties relevant to that function, and we can now transfer that fine map domain, which is illustrated on the top right. It's actually not a linear domain. It is a cluster of residues that has a very particular structural definition. And when we transfer these very particular residues identified by this method, we can indeed transfer this property from one viral backbone. And again, a first illustration of how we can possibly start manipulating and programming these particles. There's a second aspect here, which probably requires a little bit more depth, but what we're trying to do as well is build out very specific libraries that, in this case, are filtered to look at residue changes that evolution has selected. And these are illustrated in red here on this particle. And what we have done in the lab over the past couple of years is build out those types of libraries around each of these nodes or these generations of AAVs. All of these look somewhat similar to this. And that brings us to a data set of, or reagent set of about 37,000 viruses that we can interrogate in the lab along this whole lineage. And we can do basically in a higher throughput way the anecdote of the heat mapping that I just described. What we know now is that at least for one of these libraries that we have data for, 100% of these can be assembled and can be interrogated in the lab. And now we want to apply this type of reagent set to all the phenotypes that we're interested in terms of translational relevance, such as yield and manufacturing, but as well specificity questions for serological. How do we do this? We've barcoded our viruses on a one-to-one basis so that we can have a nimble NGS readout, and we can now also characterize the input versus the output, and now look for enrichment across a particular selective criteria. And I'll share with you one set of data again on manufacturing and assembly, which is when you produce with plasmids AAV virus, plasmids on the x-axis on the left here, viruses on the y-axis. In a perfect world where there's no bias and all of these produce equally well, there would be a straight diagonal across. Now we can see that certain viruses perform better. Every dot on this map is a unique viral variant. When we normalize all these data and put it in what's called an MA plot, and we basically put this diagonal flat, you can see enrichment above the 0x and de-enrichment below, meaning better producer viruses on top, worse producer on the bottom. This allows us to immediately select the better producing viruses. Great, but it gets better because we can now decouple, or we can go back and interrogate the structural diversity that we generated and see what impact it had on this particular property. And in certain cases, when we look at discrete structural toggles, there doesn't seem to be a difference between the pseudo-colored dots in yellow versus blue. But in certain cases, we see that certain particular structural motifs pull down the manufacturability and certain structural motifs increase. So in that sense, it gives us information as to how to ultimately reprogram these. And we believe that this sets us on a way in terms of particularly larger data sets generated on this. These are just kind of tweet sets. We believe it's really a big data problem to understand this very attractive viral vector system to start working towards more synthetic type vectors. If you bear with me for just a few seconds, I want to switch gears and talk about our efforts, which, by the way, Aldevron has been a supporter of, to broaden applications in gene therapy in a very different way, not through discovery, but really through collaboration. And bear with me for a few seconds here in terms of the logic that we applied to this new venture, which is a nonprofit venture that is currently primarily focused in the inherited retinal disease area, and based in part on the success of drugs like Luxturna. So on the plus side of the equation, we make a few observations that clearly there's a high promise towards developing these therapies for patients. Very similar targets are available that we can address many types of these inherited retinal diseases. In fact, there's over 200 of them that could be basically, if you will, at some level be a carbon copy of Luxturna. There's data in the literature that supports that we can indeed build those drugs. And indeed, right now, we're at a stage where we can build these therapies from a cost of goods perspective for a relatively limited amount of goods. And there are only a few patients. So we can do this. Again, this is on the plus side of it. And while this is a very active field, out of the 200 genes that we're exploring in this space, probably somewhere between 5 and 10 of those are now commercial, and the other ones are left behind. So why do we think this? Well, it's an expensive process, and it's a lengthy process to get to FDA. And unless the prevalence is there to justify to an industrial party to move this forwards, they're left behind. And on the negative side, again, and we were in this position as well, we have many proof of concept studies in the literature that aren't moving forward because they're still in search of a sponsor. And then there's an issue around drug pricing, which I'll leave. So how do we want to tackle the problem? Again, patients, academics are asking for these to be developed. We can develop those, yet they're not. What we've articulated is that once there's a prevalence below a certain threshold, basically de facto, these are commercially not viable, at least at the current. But yet we want to develop them forwards, and we want to generate economies of scale, in parts through collaborative network, where we eliminate barriers that we see as part and parcel of the for-profit. And in that, we want to establish a pre-competitive space that allows us to bring together the key expertise that is actually largely assembled here in this room to pull together in the same direction. How do we do this? Not unlike any biotech, where we really want to take a preclinical asset and bring it to what we see as the true value proposition, which is the clinical trial. We've put together an esteemed set of founders and board members, and again, and some initial funding to achieve this. And the focus, at least initially, is on inherited retinal degenerations, mainly because of a cost of goods perspective, low. These are low-dose indications, as well as the template that we have there in terms of Luxturna. What are our mission statements? We want to unlock these gene therapies for these ultra-rare type diseases that are stranded. And basically, we want to create business models where we do understand that money matters, and we want to incentivize basically the ecosystem to help us achieve this noble goal, and we think this can only be done through collaboration and creating these. And indeed, Aldevron has stepped into this mission and has supported Odelia, such as Lonza, Mass Aineer, my employer, and Patient Foundation, together with Roche and Paul. And in that, we're now moving forward, building very specific programs to the clinic through this. So with that, I went a little bit over time, but I'm happy to take any questions.
Thanks, Luke. That was some really great work. Great to hear about the progress. Talk more about that. So again, line up, Mike, if anyone has any questions. I can start one. I'm just curious with the these viral vectors you're discovering the sort of family. It's a hard problem, but could you use some of these strategies to address redosing? So if you look at it and say, maybe one dose doesn't pick half the dose, and can you sort of lay out a strategy of, we would use this one, then this one, then this one, as a way to try to maybe address those cases where we do need to redose, or is that just is what it is?
Yeah. So it is there's an analogy here between pre-existing immunity and redosing, right? Obviously, if you have a very distinct antigen, you should be able to overcome either your pre-existing immunity or the immunity that was raised based on the first injection. However, the first injection is distinct in a variety of ways from that pre-existing. One is you're in control of it, right? It happens in a clinical setting. And I would argue that there are pharmacological ways that we haven't fully understood that can manage the onset of that immunity in that B-cell immunity first setting. So I'm personally hopeful that that pharmacological regimens or other maybe B-cell depleting kind of opportunities are there to achieve exactly, because we are there at the time of that infusion. From a vector engineering perspective, I think that the opportunity is there, and similar like with pre-existing immunity, if we build a very different antigen, you will be able to redose that at least once. But then AAV generally we have seen as a very potent B-cell antigen, and to have an inert, immunologically inert AAV, I think is very tough. And again, moving far enough away from the current biology is very, so a single redosing maybe we achieve. Subsequent redosing will require multiple tools, and by the way, each of these have their own IND path. Will be very complex. But conceptually it is.
Questions? Anywhere? I had one real quick one regarding all of these variants and stuff. AAV is a pretty stable virus. Part of what? Is that still true with all these variants you're making? I mean, they're all stable, and keep that feature that I think Wilson wants for big protein. They still stable, various conditions, whatnot.
Depending to some extent on how you define stability. So one of the observations we had in this lead candidate, AncAAV, is that we saw that actually the manufacturability was somewhat down from some of the better producing viruses, such as AAV9. And our first inclination, I think, was that we were basically creating a slightly handicapped vector. Structurally something out there that created, we destabilized, therefore so we spent quite a bit of time looking at the stability of a particular particle. We found out that the inverse, at least when looking at heat stability of the particle, AncAAV was stable up to temperatures over 90 by a physical perspective, as opposed to most AAVs stable more in the range of 60 to 70. So I'm not sure whether there's a link there between the manufacturability or not, but from a biophysical perspective, we see that these.
What's your sense of when it
Yeah. I think there's quite a bit of activity in this space, and basically would come from a transformed cell line, which has been a challenge to date, mainly because of the incorporation of generally seen as a toxic gene called REP, necessary for AAV production, that is now currently transiently provided plasmid backbone or a viral. From what I can hear is that there's efforts being made that are somewhat successful to achieve this, although I haven't seen the data itself, and that would open up that continuous type. That being said, though, to support the current appetite, whether it's for larger dose applications, particularly in vivo CRISPR, as well as actually even just the Avectis AV9, these are incredibly high dose. I believe there's dramatic innovation needed in that. Right now, the innovation is indeed continuous systems, scale. Those are the two ideas that have brought to the table. I think there's biological innovation. We need to somehow get 10x productivity right now and incentivize basically those players active in manufacturing to look